Good afternoon, everybody. I hope you are well. Welcome back to a new episode of Understanding Cancer Season 2. And today we'll be discussing types of signaling pathways for normal and dysregulated BCR ABL. So what have we covered last week? So last week we discussed the TGF-beta signaling pathway, and it is a pro-inflammatory cytokine, which is a form of an immune protein that helps induce inflammation, and by doing so it can interact with proteins called SMADs. Now SMAD proteins can be regulatory, so they regulate the activity, or they work with other SMAD proteins to induce interactions, so co-mediators, or they have an inhibitory role. So, for example, some SMADs can inhibit other SMADs that regulate function. So TGF-beta binds to the C-terminal prodomain latency-related peptide, LAP, and by doing so it forms a complex called small latency complex. So in order to increase its size, what happens is it associates with TGF-beta binding protein 1, which is LTBB1, and this is what forms the larger form of the complex, which is large latent complex. So the TGF-beta ligand must be released from the large latent complex in order to be active and stimulate the SMAD downstream signaling pathway. So the TGF-beta can act as a double sword, where it can either promote tumor growth or inhibit tumor growth. So what will we learn today? So the aim of today's episode is to discuss what is the Philadelphia chromosome, the structure of BCR, the structure of ABL, how is ABL activated, cellular transduction pathway involved with BCR ABL, the link between PI3 kinase and BCR ABL, and the causes of dysregulated pathways that involves the BCR ABL. We will also discuss a brief introduction into chronic myeloid leukemia and also B-cell acute lymphoblastic leukemia. So these are the two main types of leukemia in which Philadelphia chromosome positive cells are associated with, and we will also discuss other ABL fusions that can also link to leukemia. A gentle reminder that this lecture can be divided into many sections over the course of the week, so you are able to work at your own pace and challenge yourself with a quiz. For a recap on how to support your learning, so what makes our products more unique is we provide a 5-in-1 service. So this involves providing key factor diagrams by HN Design, so these lecture notes, and it's presented in a simplified way. So that means one who, for example, the general public are able to access, and also those within institutions can also access this product. In addition, the glossary, it helps to cover any keywords that you may or may not understand. Also, we will be producing some posters. So these posters are PDF and can be printed out if needs be, for your on a wall or other aspects. And there is quizzes to test your knowledge and reflect, and there's reference lists for further reading. So if you would like to find out information regarding this topic in a further instance, then the reading list is available. I would like to give a special thanks to my parents, the rest of my family, the friends, my colleagues for the support, and also the respected teachers from undergraduate or the above who facilitated me in understanding what cancer and the signaling pathway is involved. And also special thanks to the health professions who taught me and saw the passion of oncology with me too. So what is the Philadelphia chromosome? So the Philadelphia chromosome it can be abbreviated with the term PH. So the spelling is different from the pH that is associated with the pH scale, so acidity and alkalinity, where you have a little p and a big H in representing of the hydrogen ions. But in this case, you've got a capital P and little h. So this is and cells that are do have a Philadelphia chromosome known as pH positive cells. Okay, and how it actually rises or exists is based upon a translocation between chromosome 9 and 22. So what do we mean by a translocation? That means there's a change within a gene where it's been moved. Okay, so it's been translocated from its position. So let's look more closer. So in this image we can see a normal chromosome 9 and a normal chromosome 22. Okay, and the ABL gene can be seen and the BCR gene can be seen respectively. Now when there is a translocation, there's a movement of the actual genes within the chromosome, and this is how the pH is formed, because you can see that both of the ABL and BCR gene are present within the Philadelphia, and it has some of the normal chromosome 9, and it also has some of the the chromosome 22 present in the pH. So the BCR is an abbreviation for breakpoint cluster region BCR gene, and essentially the BCR ABL produces a chimeric protein that is very tumor specific and is required for tumor proliferation and apoptosis and survival. So let's now refer onto the structure of BCR. So the structure of BCR can be seen above. So because it's a form of a protein, so you have the N terminal and the C terminal. So the N terminal is associated with the amine class, and then the C is associated with the carboxylic acid. Okay. Now the structure of BCR is divided into 4, so you have 4 domains, and it can code for 2 proteins but that vary in the molecular mass. One is 160 kD and the other one is 190 kD, so kilodaltons. So the first domain is domain 1, and that is called the dimerization motif. So dimerization motif is when it comes to, for example, substances come together, and it's on the N-terminal side. It's the coil motif. It has around 63 amino acids, and it's common to other forms of ABL. What's its role? So it helps to increase F-actin binding, and it helps to increase tyrosine kinase activity of ABL, so the phosphorylation process there. Now, the second part of the BCR structure, highlighted in blue, is a serine threonine kinase. Now, in that area, it contains a tyrosine amino acid residue called YR77, And this area is very popular by the use of adaptive proteins, which facilitate downstream signaling events. And it's also phosphorylated by the ABL tyrosine kinase. So phosphorylation is the addition of a phosphate group, and a phosphate is an example of an element that is found within the periodic table, where you've got a range of different elements there. And then the domain 3, which is in red, this is the GEF domain. So it helps to exchange between GDP and GDP, which are highly phosphorylated nucleotides there, and it's actually the central point, okay, and it contains DBL proto-oncogenic like sequences of GEF, and that helps regulate growth and differentiation. Now, DBL catalyzes the exchange of guanine nucleotides because GTP and GDP, they contain high levels of guanine. So this is how they're able to exchange the guanine nucleotides on a RAS-related molecule, such as CTC420HS. Now, the fourth part of the structure of BCR is highlighted in green, and it's the Dirac GAP. And GAP is abbreviation for GTPase activating proteins, and they particularly consist of two proteins amongst its family members, which is P21RHO and P21RAC. So they have lots of gap sequences, and the role of this is to control the rate of GTP hydrolysis of RAS proteins to the inactive form, and this is what facilitates the downstream signaling. Let us now proceed on to the structure of ABL. ABL encodes 145 kD protein, which is called P145 ABL. It's a type of enzyme, most likely the phosphokinase, so it's a non-receptor tyrosine phosphokinase, and it is situated within the cytoplasm where the chemical reactions take place. And there is low levels that are found in vitro, and it's very hard to detect in unstimulated cells. And there are two types of ABL. You have ABL and ARG, so an ABL related gene, and this is what encodes the ABL1 and ABL2 genes respectively. So here we have a structure of ABL. So similar the case, it's got N-terminal and C-terminal. The number 1, the red part, is where a lipid modification step takes place, called myristylation, and what it does, it adds a compound called myristate, and myristate has 14 carbons of fatty acids, and this myristate has been added to the N-terminal glycine residue by covalent bonding. Okay, and it was really attached to other types of proteins or amino acid residues such as lysine. The SH3, SH2, SH1, they are part of the SRC homology kinase family. SH3, SH2 are mainly involved in protein-to-protein interactions. So SH3R have an auto-inhibitory effect, where it negatively regulates the tyrosine kinase activity. So in order to do that, it needs to have the ability to react with inhibitors. In addition to that, you have SH2. So its role is to regulate the tyrosine kinase function of ABL, and it can also attach some of the targets of ABL, such as tyrosine phosphorylated proteins via arginine residues. Number 2, it's also part of the catalytic domain, and number 3, it basically has a number of parts, such as DNA binding domain, nuclear localization signals, and a binding site for actin, so it facilitates protein-to-protein interactions. So how is ABL activated? ABL activated by two processes. It's either by itself via protein mutations and deletions, or method 2, fusion of GAG, so the binding of SH3, SH2 domains to their respective ligands. So these are the two main ways in how ABL can be activated. Now the next part we're going to be discussing is the signal transduction pathways that are involved with BCR ABL. So there's a number of overlap with the other structured lessons plan for this project. For example, you have PI3K, we've got the SRC kinases, the JAK-STAT pathway, NFKB, stress-activated protein kinase, MEK kinase and ERK kinase, the Jun kinase pathway, focal adhesion kinases, mice, which is an example of a transcription factor, similar to case of NFKB, and also RAS proteins as well. But we're not going to be covering everything today. We're just going to be focusing on 3 SRC kinases, JAK-STAT pathway, and the RAS proteins. So SRC family kinases are structurally similar on a basis of that they induce tyrosine kinases activity, and they also have 4 SH domains. There are about 8 members within its family, for example PLK, FGR, FINE, HCK, LCK, LIN, CSR, C, and S. So BCR ABL can activate LIN and HCK in myeloid cell lines. So SRC kinases may be associated indirectly to BCR ABL. This is through molecules such as protein tyrosine phosphatases. So in addition to that, it's important to say that even though they partake in this, the activation of CRC kinases is not dependent on BCR ABL only. Now with STAT5 JAK2, so STAT is the abbreviation for signal transducers and activators of transcription, and what it does is that it links with cytokine receptor stimulation to induce transcription, and it can be found in BCR ABL cells. And this is what induces leukemogenesis, leukemia, especially some of the STAT proteins such as STAT1, STAT5, and STAT6. So what actually happens is that there's a cytokine receptor stimulation. The tyrosine kinase protein phosphorylates by JAK family kinases. The STAT protein dimerizes, so it becomes a dimer, and then the STAT translocated to the nucleus to bind to specific DNA elements to induce transcriptions. Now RAS proteins are very important. So you saw their importance within EGFR, with GPCR. So they are heavily involved in terms of signal transduction and the cellular response, for example, proliferation and malignant transformation. So RAS proteins are able to transduce the signals that are regulated by either GAPs or GEFs. So GAPs, they enhance the rate of GTP hydrolysis to GDP. So it essentially negatively regulates the RAS function. Alternatively, GEF, they do induce exchange of GDP for GTP and RAS, and it positively regulates RAS function. So how does RAS get activated via BCR? So there is an adapter called GRB2 that is linked with the SH2 domain to the conserved tyrosine residue via R77 within BCR. Okay, now because of this binding, it helps recruit SOS, which stimulates RAS, and RAS can also be activated by inhibiting GAP, and also be activated by CRKL via SOS. So let's have more details about CRKL. So CRKL is an adapter protein that creates a link between several ABL forms, for example P210 BCR ABL, and it also influences the focal adhesion molecules such as FAK and actin and paxillin. Okay, so it can be undergo phosphorylated via its tyrosine kinase domain in cells that contains the Philadelphia chromosome. It also binds to proline amino acid residues in the BCR ABL, and it can stimulate RAS. Okay, so that could be by SOS or by CR3G. So CR3G, CRKSH3 domain binding guanine nucleotide releasing factor is also associated with splicing of the DNA, and also the there's another one, yes, embryonic development. So what is the link between the PI3 kinase and BCR ABL? The link between PI3 kinase and BCR ABL is that the PI3 kinase, as you should know already from the previous lecture, is that it contains P85 regulatory subunits and P110 catalytic domain via its enzymatic activity. So the crosstalk that occurs between them is through a protein called casistas B lineage lymphoma protein, and it's an example of a proto-oncogene, which means that it's a normal gene, but when it's mutated, it can cause cancer and become a proto-oncogene. So CBL is an example of a proto-oncogene. CML is an example of a proto-oncogene, and that is facilitated in BCR ABL expressing cells via its tyrosine residues. So BCR ABL are associated with apoptosis. How? It can either promote or it can evade. So if you want to promote apoptosis, it can be done by the BCL family of proteins, more particularly the BCL2, BCLXL. Now, to be able to evade apoptosis, which is one of the core hallmarks of cancer, especially in disease states or normal states of chronic myeloid leukemia, there is a downregulation of an inhibitor called P27. So what it does, it inhibits the kinase so there will be less phosphorylation taking place. So what are the causes of dysregulated cancer pathways? It can be associated with mutations, for example, defects in the SH2 domain. It can influence protein interaction and lowers phosphotyrosine kinase, okay, binding. Another example is a direct interaction with retinoblastoma. So retinoblastoma is an example of a tumor suppressor gene, and when it can also increase ABL expression, And there is another type of tumor suppressor. So it's P53, otherwise known as the guardian of the genome, and it directly interacts with P53 by overexpressing ABL. So let's now proceed on to the several examples of disease states, such as chronic myeloid leukemia. So, chronic myeloid leukemia is a result of what we say, hemopoietic pluripotent stem cells. So that means that the pluripotent, that the cells are still in an immature state, okay, and it can see, as the name suggests, it mainly affects the myeloids that is present within the bone marrow. So this is inside the bone where you have the spongy and the compact areas, and it can affect the myeloid that's involved within the white blood cell precursors called myeloblasts, and that can be further differentiated to form granulocytes. So the CML can begin at a chronic stage and then can progress into a terminal blastic phase. And many of the ways in how it can be treated is by inhibiting tyrosine kinase. One example is imatinib or Gleevec, and what it does is that rather than completely eliminating what it does is partially eliminates BCR ABL and expressing leukemia cells. In addition to that, due to the current status, it can help develop drug resistance. So also a study have suggested that imatinib inhibited BCR ABL, and it can still induce SRC kinases, and this is results in another type called acute lymphoblastic leukemia. So lymphoblastic is another term of a cell that has a normal abnormal shape and influences its function. So if we look at a blood stem cell, so stem cells, these are undifferentiated cells that are self-renewal, and they differentiate to form specialized cells with function. Now within the blood stem cells you have two types. You have the myeloid and the lymphoid. The myeloid can assist to produce red blood cells. They also have a protein involved in red blood cells called erythropoietin, and the myeloid stem cells are also associated with platelets, so these are small fragments to be utilized. And there's the myeloblast, which we discussed earlier, and it can differentiate into forming granulocytes. And you can see that the granulocytes, they vary in color. So you have the basophils, so the xenophils, and the neutrophils. So this may be associated in terms of the staining. Now the other type of blood stem cell is lymphoid stem cells. So you have primary lymphoid areas and secondary lymphoid areas. So the lymphoblast itself is an example of an abnormal white blood cell, and it can be subdivided into B lymphocytes or B cells, T lymphocytes or T cells, or natural killer cells. Okay, so whether the white blood cells are from a lymphoblast or whether the white blood cells are from myeloblast, collectively they are known as the white blood cells. Now, in terms of the onset or the prognosis, it mainly affects people of middle-aged of any form of gender, and it's between 40 to 60 years of age. And common symptoms that can occur is fever or loss of weight, or if there's sweating or anemia. So there's a variety of different types of anemia. It can be associated with a lack of iron and low hemoglobin levels. Can be associated by lack of vitamin B12, so folic acid. Also, it can be linked to having a precocious anemia. So that is when the absorption of vitamin B12 is not sufficiently working within the intestines there. So this image is a microscopic image, and one may think that the red blood cells are going to be in the red area and so forth. But via this light micrograph of blood cells of the bone marrow, the pink cells actually represent the white blood cell precursors, so the myeloblasts or the white blood cells, and the orange represents the red blood cells. Now P62DOK is an example of another adaptive protein of BCR ABL, and many of the chronic myeloid leukemia patients have this form of protein, and it links with RAS-GAP, and this inhibits the phosphorylation activity, and it has the ability to form complexes with SHIP1 in hematopoietic cells that express BCR ABL. So let us now proceed with the B-cell acute lymphoblastic leukemia. So this is more of a childhood cancer, and the cause is not really known, but it's more associated with inherited or genetic or congenital conditions where children aged under 15 years are exposed to this. However, they were able to have increased survival rate in comparison to other types. And amongst the symptoms is anemia, bleeding, infection, and hepatosplenomegaly. So with that phrase there, that's when there is an increase in size of the area there, especially, for example, in the liver and the spleen area. Other APL fusions. Now, besides the BCR ABL fusion, there is also NUP214 ABL, and this is more associated with T-cell acute lymphoblastic leukemia, where it mainly affects 7%, and it's on a long period of time. Now, the first exon of ABL is missing in the protein. So what that does, it can cause an amplification, so a copy, where there's an extra copy of the chromosome, and that is present within the long arm of the chromosome 9, and this fuses with most of the with the NUP214 exons in ABL1, and this can also influence how effective the drugs are. So we have reached the end of today's episode. So the PH chromosome arises from a translocation between chromosomes 9 and 22, and this affects the ABL and BCR gene. There are two main cancers associated with ABL-BCR translocation. So there's chronic myeloid leukemia that mainly affects middle-aged patients, and also B-cell acute lymphoblastic leukemia. This mainly affects the pediatric patients under 15 years of age. So there are other ABL fusions that can affect or cause other types of leukemia. RAS is the main sickle transduction pathway that is associated with BCR ABL, and it can be activated by various ways. So it can be bound to the GRB2, can be bound to the BCR. It can also be recruited by SRS to stimulate RAS. It can also be activated by inhibiting GAP or activated by CRKL via the SRS. So here's a reference reading for further reading. Next week we will be discussing the dysregulated pathway of Notch and also the normal pathway mechanism. I hope you have benefited from this lecture. Stay tuned to next week. Thank you very much. Take care.